Hey everyone, welcome to Chapter 5. Before we get started, a couple quick announcements. So Chapter 5 is large. There are a lot of pages in this chapter. There are several topics. And before you get too carried away, I want to just let everybody know that we are only going to be covering part of section 5.1. So 5.1 is protein function as applied to the story of hemoglobin and myoglobin. So two proteins that carry oxygen, and we're going to look at how amino acid sequence affects structure and how structure affects function with those two particular proteins. Myoglobin and hemoglobin were the first two proteins to have their three-dimensional coordinates determined by X-ray crystallography. And they're probably two of the most widely studied and most understood proteins out there. So that's why they make great case study proteins to look at. So let's get started. So the first thing that we want to talk about, right, we need to introduce this concept of proteins binding to ligands. Ligands is a key word that we're going to use, and basically we're talking about a big molecule, a protein, binding to a smaller molecule, a ligand. And in Chapter 6, when we start talking about enzymes, we'll use the term ligand or substrate kind of interchangeably. They both mean the same thing. But for now, we're just going to go with ligand. So we need to talk about how proteins and other molecules interact with each other. We're going to set up a little bit of mathematical framework based on some Gen Chem equilibrium constants. And the reason that we're going to do that is so that we can look at these graphs of how proteins interact with small molecules, and particularly how strong that interaction is. So I don't really care too much about the math behind it, but the graph itself and what it tells us is actually super useful and pretty important. And then we'll get to the interaction of the family of globins, which, like I said, includes myoglobin and hemoglobin, how they interact with oxygen and non-oxygen molecules, and which amino acids are involved, and all kinds of cool stuff. Okay, so the first thing that we have to discuss here is proteins are not the rigid, static structures that you see in the textbook. So if you look in the textbook, or if you look in the PDB, the protein data bank, you can see many, many structures of proteins, and they are these nice, perfect, here's an alpha helix, here's a beta sheet. It doesn't look like there's any movement involved in those structures. Well, that couldn't be further from the truth, actually. When you take a protein and put it into a solution, there are lots of internal motions that are activated while the protein is dissolved in the solution. And this is something that we ignore because honestly it's hard to illustrate. But you can have dynamics or protein movements that occur on timescales down to, let's say, 10 to the minus 12 per second, the whole way up to maybe something on the order of seconds or hours or days even. Some of these things are very, very related to how the protein functions. Some of these things are just, I guess, side effects of how stable the protein is in solution. So these motions, sometimes we refer to the protein as breathing. And if you think of kind of a spherical protein, when I say that the protein breathes, you can imagine that sphere kind of getting bigger and getting smaller and getting bigger and getting smaller. Just like if you were watching someone breathe, if you were watching their chest move up and down, okay? Those kind of motions are important because there are proteins that bind ligands inside. And the only way for the ligand to get inside is to go in as this protein is opening up as it breathes. And then when it closes back down, the opening is gone. Okay, so proteins are dynamic. They have to move. It's part of their function, and it's important. So another important thing is right now we are talking about reversible binding of ligands. This is super important and essential for what we want to talk about. Just to give you a very quick example, imagine myoglobin or hemoglobin. Imagine if they went to your lungs, picked up oxygen, and then went out into the rest of your body to drop that oxygen molecule off to the organs and tissues that needed it. But imagine that this was a permanent binding event, right? There is no unbinding, there is no dissociation. That means that every molecule of heme or molecule of hemoglobin that was carrying oxygen wouldn't be able to release it to the tissues that actually needed it. So it's important that we talk about reversible binding. This is a very important area of biochemistry, of protein interactions, of molecular interactions. We can use this for things like cell signaling. We can use it for the obvious that we're talking about now, which is myoglobin and hemoglobin and respiration and moving oxygen around in your body. Hormone receptors, the immune system, muscle movements, all of these things rely on reversible binding of ligands. So we have to make sure that we understand proteins are dynamic, number one, and number two, binding processes in this case are reversible. Okay. So let's set up our system first. We have a reversible transient process. It's a chemical reaction. That means that it's an equilibrium reaction, right? It's reversible, so it exists in equilibrium. We have our reactants, A and B, and we have our products, AB. So A and B in this case would be oxygen and myoglobin, or oxygen and hemoglobin. So we take those two things, oxygen and hemoglobin, add them together, and we get an oxygen hemoglobin complex. That's AB. That process is reversible, so those two things can dissociate and give us back our individual molecules of oxygen and myoglobin or hemoglobin. There's a specific place where this binding event takes place, and that's called the binding site. In an enzyme that we'll talk about in Chapter 6, the binding site is going to be where the reaction happens. In Chapter 5, the binding site is somewhere on the surface of the protein where the molecular interaction between protein and ligand takes place. If you asked me, and if I had to generalize, I would say that most proteins have one binding site per molecule. That's not universally true. There are some other cases where there's some duplicated motifs or duplicated regions of the protein that look very similar, and in that case the binding site is also duplicated. But like I said, most cases you get one binding site per protein. And not surprisingly, like I said, these binding reactions are reversible. There is no covalent modification to either molecule. So that means that there are no bonds formed and there are no bonds broken. So when we're talking about these interactions, we're talking about the same four non-covalent interactions that dictated how protein structure came to be in the previous chapters, right? So we're talking about the hydrophobic effect, we're talking about ionic interactions, hydrogen bonds, dipole-dipole interactions, and van der Waals forces.